Thank you, sir.
Okay.
So I will start. Welcome all the participants for today's webinar, which is very interesting because we are again touching plant-based pharmaceutical solutions. So on behalf of Kairos Kinetic and Global Biomedical Research Foundation, I welcome all of you for today's webinar, and we are really honored that Professor Dr. P. Palani from Center for Advanced Studies in Botany, University of Madras, Guindy campus, accepted to be our speaker for this week. Thank you, sir, and welcome once again to our webinar. I'll just briefly introduce for the benefit of our audience, very quick update about Sir's achievements and background because I wanted him to talk and interact with you. So I'll save a little bit of time, but I'll be very quick. Sir has obtained his PhD in 2002 in from Center for Advanced Studies in Botany. That's a very rare achievement. He achieved his PhD, and currently, sir is also heading there as a professor. Also, sir obtained MPhil in Life Sciences from School of Life Sciences, Bharati Dasan University in 1993, and his area of specialization again plant tissue culture. Obtained MS Botany from Government Arts College, Nilagiri District, Coimbatore. So that's very unique reporter Chennai, Tiruchirappalli, as well as Coimbatore. And Sir has great achievements and academic honors for his research related to the topics today we are discussing. And Sir also has experience at national and international laboratories and 17 years of teaching experience at master's level. Coming to academic honors and awards, Sir is appointed as Member Secretary, Institutional Animal Ethics Committee for University of Madras, Guindy from 2019 till date, and Best Researcher Award by University of Madras for the year 2019 and 2020. So also, Sir received Dr. Kalyana Sundaram Memorial Award Prize for the Best PhD Thesis. That's so wonderful in 2003. And Sir also has a lot of fellowships under his credit, including postdoctoral research fellowship at NIH, Texas, senior research fellowship UGC India, as well as junior research fellowship UGC India. That's a quite an achievement, sir. Sir, you have visited NIH campus. That's great.
Yes.
We always read the articles, but I never imagined like any given day I will be visiting the campus now with all this latest politics. But yeah, that's a great thing. Congratulations for such a great achievement, sir. And sir has also received research projects as latest as in 2020, development of drugs from natural resources for
ಮಂಜೆಲ್ಲ ವ್ಯಾಪನೆ ಮಾಡೋದು
Kannan sir, can you mute the participant? Someone is having some background noise. Yes. I'll repeat it again. Sir received almost like 14 lakhs grant in October 2020 for development of drugs from natural resources for biomedical application, RUSA 2.0. And Sir has also represented in wonderful international and national conferences, delivered multiple lectures and talks, and Sir's H index is Google Scholar 20 and Scopus 16. Currently, Sir is also guiding PDFs, that is postdoc fellowships. 3 candidates, PhD degrees already awarded under his guidance were 12. Currently working with 6 PhD students. And Sir also contributed for achieving MPhil degree by candidates around 37 numbers. With that very quick review of Sir's entire biography and career achievements, I request once again, Sir, that we welcome you for today's webinar and kindly proceed to your topic, sir.
Yes, madam. Thank you very much, and very good evening to all the participating members of this webinar. Uh, Today, I will be talking a little bit about the research work that has been being carried out in my laboratory, and
Can
so
share the screen, sir,
yes,
if you
yes,
have. Yes, sir.
I,
Yes, sir.
yeah. Can you tell me whether the slide is visible?
Yes, sir. Slide set is visible. You can move on to slideshow.
Yeah. I put it in slideshow only.
But okay, for us it is showing all the slides on the left side. Maybe some settings problem, but no problem. Then you can proceed with the presentation, sir.
Okay. So in my screen, it appears full screen.
Okay, that's the. I think so. Okay, fine, sir. Then
Okay.
you can please
So
proceed.
today's topic on which I'm going to talk about is development of anti-candidal drugs from plants. So I have given my contact address and email ID. If anyone wants to contact me after this lecture, you are very welcome. And now I go to the lecture part of the webinar. So we have to ask a question: why we have to develop a drug from plants, and what is the problem with the drugs, synthetic drugs, which are already available for controlling and curing the diseases caused by one pathogenic fungus called Candida albicans. And you know that the species belonging to the genus Candida, they are most of the species are opportunistic pathogens.
I would like to excuse, sir. In my screen, I'm not seeing the second slide, but if everyone else is able to see that, I just want them to confirm because are you in this first slide, sir?
No, I'm in the second slide.
Okay. So I
Yes, sir. I think we are stuck with the slides, sir.
uh, I
It's
think
in the
there may
first
be
slide, and
some
it is not in
slide.
slideshow.
Okay,
Yeah.
now what I will do, I will close it
Yes,
and
sir.
open.
Sure,
Okay.
sir. No
Now
problem.
it is seen, but it is not in slideshow.
You yes, you can restart, sir. Meanwhile, so that we will try once again.
Okay.
Yeah. So meanwhile, we welcome all the audience once again, and please post your questions in the chat box if you are interested to interact with our professor at the end of the session. And I'll be moderating the session. So in between, when sir is presenting, if you have any questions, kindly post it there. Anyway, we will also open up the discussion after the session, so you can also interact. Sir, you can proceed with sharing.
Yeah. Just
Yes,
sharing one more time.
yes, sir, please.
So now, are you able to see the full screen?
Not personally, I'm not able to see, sir, but I'm able to see the presentation on the first slide.
Okay, I'm in the first slide only, but
Okay.
I don't know what is the problem. But in my
We,
screen, everything is
we
full.
experienced earlier also this kind of issue, sir. It's not a problem. You can change the next slide, and if that is visible, we can proceed, sir.
Okay,
Srinivasan,
so now are you able to see this?
sir, our participant Srinivasan has offered that
I'm in the second.
you can press the F5 button and check, sir. He says like you can check.
No, yes, Sarah's come to his slideshow mode in his system, but it is not visible for us. Yes, now
Sir,
same.
your your voices are breaking. I'm not getting your points clearly.
Yes, sir. Maybe net is. Net speed is an issue, sir.
Okay.
Maybe you can share the slides to me, sir, so that I can. You can
Yeah,
just
that
post me
also
my
will
mail.
work. Yeah, that also will work. I will
I
send.
will change the slides for you, sir.
Yeah.
We are able to see the second slide.
Okay.
So either you can proceed like this, or if you prefer, you can send it out to
Now, this slide is
we request the participants to
the
mute.
third one. Can you see the third one?
Yes.
Okay, so it works very well with the full screen, not with the full screen mode, but it works with full screen mode.
Okay, so then you can proceed from the second slide itself and we will see.
Yeah.
Yes, sir.
So this, I asked a question: what is the necessity to develop a drug from plant sources rather than the drugs which are already available commercially in the market to cure so many diseases caused by Candida albicans? So in this particular slide, what you see here is the various diseases caused by Candida, species of Candida albicans. So it causes vaginitis and it causes oral candidiasis, cutaneous candidiasis, candidemia, and systemic infections. And I told you before that Candida albicans is an opportunistic pathogen, and every human being has Candida albicans either in the oral cavity or in the vaginal cavity, and it remains as a normal flora. But when the immune system goes down, the same organism becomes a pathogenic organism and causes these many diseases. And one more characteristics associated with the Candida albicans that it is a multimorphic fungus and which can exist in the form of yeast cells. It can also produce germ tubes, and the germ tube finally will develop into mycelia. And it can also exist in the form of a pseudohyphae. And these are the different morphological characteristics of Candida albicans. More importantly, it has the capacity to form biofilm. And virulent factors. I must also tell you that Candida albicans is a unicellular fungus, and if the environmental conditions permits, it produces germ tube and mycelia. And the production of mycelia or a germ tube is also considered as one of the virulence factors. And the environmental conditions actually favors the production of germ tube and formation of biofilm. Again, the immune system of the particular individual favors the production of germ tube and the production of these virulent factors, and ultimately causes various diseases. And here are some of the species of Candida. And among the Candida species, Candida albicans is the notorious species responsible for various diseases in human being. And other than Candida albicans, we have Candida parapsilosis, Candida tropicalis, Candida krusei, Candida glabrata, Candida guillermondiae, Candida arthroxylosis, metopsilosis, Femata, and Lusitania. And these species of Candida have been recently reported from the various diseases. And on top of it, we have recently encountering the occurrence of infection caused by Candida auris. And in this picture, The areas which have been highlighted in blue color are the regions where the Candida auris infection have been reported in recent times. So these are some of the morphological forms of a Candida. The first one is the yeast cells, which primarily exist in the form of yeast, and they produce a pseudohyphae, and they also produce germ tubes. And the production of pseudohyphae and hyphae are considered as virulence factors, in addition to formation of biofilm. And here are the conventionally used drugs. And we have three different classes of drugs, polyenes, echinocandins, and azoles. So under polyenes, we have the most commonly used anticandidal drug is amphotericin B. And under the azole category, the most commonly used azole drug is fluconazole. In addition to the fluconazole, we have other variants of azole drugs like itraconazole, ketoconazole, and miconazole, and clatrimazole. And echinocandins, and these are the newly developed drugs. And caspofungin is the most commonly used echinocandin. And we have received so much of information about caspofungin. Caspofungin is the drug which target the glucan synthesis, and thereby the fungus will be deprived of glucan, and the growth will be prevented. And what is very important here is, so caspofungin is the newly developed drug, and it has been reported to show potent activity against various species of Candida, including the Candida albicans. But there is a recent review, I would say that is research work published by one laboratory in Tamil Nadu. And they have isolated Candida auris from the infected patients, the coronavirus infected patients. And these Candida auris species has been found to be resistant even to caspofungin. So when it is resistant to caspofungin, it should be also resistant to most of the fluconazole drug and even polyenes like amphotericin B. So the point I would like to inform you that we have a large number of anticandidal drugs, but the problem is most of the species of Candida and Candida albicans, they develop resistance against these anticandidal drugs. So therefore, we have a bigger challenge of finding an alternative anticandidal drugs from other sources like plants. So in this respect, here are some of the mechanism of action of some of the conventionally used anticandidal drugs. And here we have the echinocandins, and they prevent the synthesis of glucan in the cell wall. And we have another anticandidal drug called flucytosine. And these particular anticandidal drugs inhibit the synthesis of nucleic acid in the cell. And we have the chrysofulvin, which primarily inhibits the formation of microtubule in the plasma membrane. And we have azoles, allylamines, and the thiocarbamates, and they primarily target the ergosterol biosynthetic pathway. And then polyene, as you know, that it binds the ergosterol in the plasma membrane, and this disrupt the membrane integrity. So these are the different mechanism of action of some of the anticandidal drugs. And a while ago, I just described about the difficulties we have with the conventionally used anticandidal drugs. So one of the major problems that we encounter with the available anticandidal drugs is that most of the species of Candida, including Candida albicans, they develop resistance. And once they develop resistance, it would not be possible to cure or to kill them with any available anticandidal drugs. So therefore, we have a big challenge of identifying and looking for an alternative drug, which is more potent than any available anticandidal drugs. So in this respect, we set out a research to identify an anticandidal molecule from a plant source. And before we could undertake the research, and we want to isolate the Candida, the pathogenic Candida species from clinical samples. And with the help of the support provided by SRM Tyndall College in Ramavaram, we were able to isolate the clinical samples from infected patients. And we were able to isolate 11 clinical strains of Candida. And one of the primary requirement is we wanted to identify what species are they. And we used a specific medium called chrome agar. And when you grow Candida species on the medium containing chrome agar, and different species will form different colored colonies based on the color of the colonies. And we were, to a certain extent, could identify what species of Candida it is. So likewise, so total number of clinical isolates we isolated is 11. And out of 11 different samples, 10 of them happen to be Candida albicans. And one was Candida tropicalis. And the next thing, what we have done is, we wanted to study the resistant profiles of all these Candida species. And this resistance profile was tested against one particular drug, which is most commonly used for determining the resistant pattern of Candida and Candida albicans. And this is how we prepared the drug. And we did the test by using the microdilution method as has been prescribed by CLSI. And we took 24-hour old culture as inoculum. And the cell density was adjusted to 5 times 10 to the power minus 3 per ml. And then the cells were inoculated to the medium containing fluconazole. And the concentration range we used was 0.01 microgram per ml to 128 microgram per ml. And we grew the samples for 24 hours. And we determined the susceptibility pattern. So here are the susceptibility profile of different Candida albicans strains and Candida tropicalis. And on top of the table, you have the strain names, which has been indicated as A1, A2 till A11. So A11 is actually Candida parapsilosis, Candida tropicalis, sorry, Candida tropicalis. And the strains from A1 to A10, all of them are different strains of Candida albicans. And if you look at the resistance pattern, and A8, Candida albicans strain A8, A9, A10, and Candida tropicalis, which is A11, and all of them showed resistance against fluconazole. Whereas the strains A1 to A7, they are either moderately resistant or sometimes they were susceptible also. So what we could find out of 11 different clinical strains we isolated, and 4 of them were found to be highly resistant to fluconazole, and we categorized them as fluconazole resistant isolates. And thereafter, we set out to isolate and identify one particular compound from a plant source. And so this is actually the plant source. This plant is, the common name of this plant is hairy spurge. And the botanical name of this plant is called Euphorbia crypta. And the isolation of anticandidal compound from this particular plant was accomplished with the leaf sample. So we collected a large quantity of leaf sample, and the samples were dried under room temperature, and the dried leaf were powdered, and the powdered samples was extracted with 4 different organic solvents like hexane, ethyl acetate, acetone, and ethanol. And in addition, we also got extract with water, and that extract is aqueous extract. So a total number of 5 different extracts were obtained, and we wanted to see in which organic solvent the anticandidal compound we are getting in a relatively high quantity. So that was the idea why we have used several organic solvents. And we used the rotary evaporator instrument to get the extract. The powdered sample was mixed with one particular solvent, and it was boiled in this particular instrument, and we got the extract. And the extract was finally concentrated either at the room temperature just simply by evaporating the solvent, and the concentrated extract was used for further experiment. And so here are the results obtained with the anticandidal activity of a hexane extract obtained from the chosen plant. And here are the Candida strains, C1, C1 to C11, or the clinical strains of Candida species. And in one particular C1, I would like to say that the anticandidal activity was primarily evaluated using the well diffusion method. I hope that majority of you will be knowing what is a well diffusion assay for anticandidal or antibacterial activity. So in this case, the middle well, which has been indicated as A, was the known anticandidal drug. We used amphotericin B as the positive control, and B is the chemist. DMS, and well C is 250 microgram of extract obtained with hexane, and D is 500 microgram, and well E is 1 milligram crude extract obtained from hexane extract. Similarly, the pattern of treatment in all other strains was similar to this one. So the middle well in all the cases is a positive control, and B is the DMSO control, and C is 250 microgram, D is 500, and E is 1 milligram per ml. And you can clearly see, even with the crude extract, we could observe a significant inhibition of the growth. And similarly, and this anticandidal activity was observed against all other strains. In some cases, we got the pronounced anticandidal activity by looking at the extent of lysis or extent of inhibition. And this was the case against all other Candida species. And now we wanted to purify the compound which is responsible for the anticandidal activity. We wanted to purify from the crude extract obtained from hexane. And so here are the results we obtained with extracts obtained from different organic solvents like hexane, ethyl acetate, acetone, ethanol, and aqueous extract. As you can see here, the extract obtained with the hexane, they showed relatively increased anticandidal activity against all the clinical strains, followed by the crude extract obtained with ethyl acetate. And among the 4 different organic solvent extract we got, the extract obtained with hexane showed more pronounced activity, anticandidal activity against all the clinical strains, and therefore the subsequent experiments were conducted only with the hexane extract. And this was the assay we performed, and which is called the XTD assay. And we wanted to see the minimum inhibitory concentration of the crude extract obtained with the hexane. So though we isolated 11 different clinical isolates, and we used only 2 strains, C11 and C12, and C11 is a susceptible strain, and C12 was a resistant, fluconazole resistant strain. And we wanted to determine the minimum inhibitory concentration of crude extract against these strains of Candida albicans. So as you can see here, so the orange color is due to the formation of a product called pharmazone. When the cells are alive, they produce mitochondrial dehydrogenase enzyme, and this enzyme will react with the substrate available in the reaction mixture, and the orange color development will occur. So wherever you see orange color development, there you get the growth of the organism, and wherever you don't see the orange color development, and those are the wells in which the Candida growth has been inhibited. So this is the way we did this is called the XTT assay, and we found out the minimum inhibitory concentration. The minimum inhibitory concentration for strain C11 was found to be 0.25 microgram per ml. Similarly, we also found out the minimum inhibitory concentration for the resistant strain, and for the resistant strain, the quantity of the crude extract was 1 microgram per ml. So this was the minimum inhibitory concentration we determined against the susceptible and resistant isolates of candida albicans. And then we proceeded to purify and characterize the anticandidal compound, which is responsible for killing the organism. So what we did, so we have done the HPTLC analysis with the crude extract. And here are the banding pattern of compounds that has been developed with the HPTLC technique. And these three pictures are not different one, but the same picture, but under different lights. So this one was viewed under visible light, and the red bands are viewed under UV light at 254 nanometer, and the green one was viewed under fluorescence light at 366 nanometer. And all these compounds were developed in TLC using a mobile system consisting of hexane, ethyl acetate, acetone in the ratio of 8:1:1. So that was the mobile phase we used to develop all the compounds in the TLC plate, and they were viewed under different lights. So these are the banding patterns we obtained from the crude extract, obtained with hexane. And then we gone for the next purification technique like column chromatographic separation. So in this is actually the chromatographic column. The silica gel column has been packed in the glass column, and we loaded our sample. And then the mobile phase consisted of hexane, ethyl acetate, acetone, 8:1:1 ratio. And then the sample was loaded, and the mobile phase was continuously supplied. And at the end of the column tube, we were able to collect fractions. And as many as 160 fractions, and each fraction containing 5 ml volume of the sample we collected. And all the fractions were subjected to HPTLC analysis. So a small volume of sample from each fractions. So the total number of fractions are 160. And from each fraction, we took a small volume of sample, and that amount of sample was used for HPTLC analysis. And after development, the TLC plates were looked at under various lights. So these are the compounds that we observed in fractions 1 to 5. So we have 15 different wells in one TLC plate. Of course, in some cases, the bands are clearly visible. In other cases, the bands are not visible. And so is the case with another light source, UV light 254 nanometer. In some cases, you see the presence of separated compounds. In other cases, in other fractions, you don't have the compounds. So that indicates that some of the fractions do not have the separated compounds. And here are the fractions. HPTLC analysis of fractions 16 to 31. And you get some of the separated compounds here. Similarly, we have done the HPTLC analysis for all the 160 fractions. And not only that, we also performed the XTT analysis. And the first plate has been loaded with the samples of a fraction 1 to 94. So number 1 indicates this is a fraction, the sample we loaded from a fraction number 1. And similarly, so 94 samples have been loaded in a single plate, and we did the XTT assay. As I have explained before, wherever you see the orange color development, and these are the fractions where you don't have the anticandidal. And wherever you see no development of orange color, and those are the fractions where you have anticandidal drugs. And in fractions of 95 to 160, except 1 or 2 fractions, all other fractions inhibited the growth of candida albicans. So that means most of the 160 fractions, they have compound, 1 or other compounds, and they inhibit the growth of candida albicans. And finally, so there may be, the plant may have several compounds that may show anticandidal activity, but we want to purify and characterize one particular compound that shows more potent activity against candida albicans. So that was our objective. And then, so here are the banding pattern of crude extract. The first lane is the banding pattern of crude extract, and we named the bands with the serial numbers. So the lowermost band was 1, serial number 1, number 2, number 3, 4, 5, 6, 7, and 8. So we have given serial number to the bands, and what we have done next, and we also analyzed we subjected the column eluted fractions also for HPTLC analysis. And here the figure B is the chromatogram that has been developed with fooled fractions. Fooled fractions after after the silica gel column chromatography. So what we have done here is we took one fooled fraction that shows relatively higher anticandidal activity, and we wanted to see the banding pattern of that fraction. And fortunately, we got 4 different bands. The upper band was given serial number 1, and the next one was named 2, 3, and 4. And we wanted to see which of the 4 compound actually shows anticandidal activity. What we did, we eluted each of the compound individually from the silica gel plate directly. So we scraped this particular area from the TLC plate, and the compound present in the scraped portion of the TLC plate was solubilized in solvent, and that solvent was loaded in the well diffusion method. So well 1 is the compound 1 isolated from the HPTLC chromatogram. Similarly, and we have the band number 2. That band number 2 was scraped, and the scraped compound was loaded in the well 2 in the Petri plate. And the 3, so this is the 3, and this is the 4th. We wanted to see which of the 4 bands shows potent anticandidal activity. But fortunately, we got compound number 1 that showed pronounced anticandidal activity. And so this compound were further taken for purification. And before we could complete the purification part, we wanted to also analyze the crude extract with GCMS technique. We performed GCMS analysis with crude extract, and we got 5 different major peaks. And one of the peaks, that is azulene, the second one, the azulene band was relatively higher. And we got this particular band, particular compound at a retention time of 8.176. And the percentage of area was found to be 51.17. And when we compare the amount of molecule present, so azulene was found to be relatively higher when compared to all other compounds. So we thought that azulene could be the potent anticandidal drugs. So what we did, we removed, in the second level of experiment, we removed each of the bands. So the bottommost one is band number 1, 2, 3, 4, 5, 6, 7, 8. And we removed them individually. And we did XTT assay to see which of the bands has a more pronounced anticandidal activity. Unfortunately, all the compounds, so band number 1, band number 4, band number 5, 6, 8, all of them showed anticandidal activity. So we were a little bit confused. So which of the compound we want to purify? If all more than 5 different bands show anticandidal activity, so it would be difficult to identify which compound has a more pronounced anticandidal activity. So what we did, so we took individual bands. So these are the individual bands. So this particular band was isolated, and then we did another HPTLC analysis, and we got only a single band that has been scraped from band number 1. But when we scraped band number 2, this is the band number 2, and we separated this particular band again on HPTLC, and to our surprise, we found that so whatever appeared as a single compound in the previous chromatogram now has been separated into 4 different compounds. A single compound, which was earlier in a single, which was in the chromatogram, now we analyzed this again on HPTLC chromatogram, and we got 4 different bands. Similarly, band number 5 from this, so this is band number 5, and this was scraped and again run on a TLC, and this in turn has resulted in the separation of 3 other bands. So the sixth band we scraped from this chromatogram was again run on another TLC plate, and this has resulted in the separation of 3 other compounds. Likewise, the compound, the band number 8 from here also separated into 3 other compounds. And after this, we evaluated anticandidal activity of all the separated bands. So here, so this is the band number 1. So we have a single band, and this single band was included in well number 1. And 4.1 is this one. So 4.1, this is the compound, and this compound was loaded in the second well. and the second well also has a Candida albicans. So the 4.1 band does not show any anticandidal activity, and therefore the Candida normally grows, and it produces mitochondrial dehydrogenase enzyme, and which reacts with the XTD, and therefore the orange color development occurs. But if you see band number 4.2 and 4.3, and these two bands, 4.2 and 4.3 is here, 4.2 is here, and 4.3 is here. And these two compounds completely inhibited the growth of Candida albicans. And we have three different bands from fifth band. That fifth band has now resolved into three bands, and only one of them, 5.1, has inhibited the growth of Candida. And the other two bands, 5.2 and 5.3, they have not inhibited the growth of Candida albicans. And so what is very important here, and the sixth band was re-chromatographed, and which has resolved into three compounds, and all the three compounds inhibited the growth of the organism. So from this experiment, what information we got was, among the different bands that showed anticandidal activity, the bands, so the sub-bands 6.1, 6.2, 6.3, and they showed relatively higher inhibitory activity when compared to all other bands. So we took this sample and we gone for further purification. So here are the bands 6.1 and 6.2 and 6.3, and we took the third band, 6.3 band, and which was re-chromatographed on another TLC plate, and this single band now has resolved into three other bands, and we named it as 631, 632, 633, and we scraped them individually and evaluated against Candida albicans. So here the first one is a 6.631. All the samples are a 6.31, and the second column is 632, and the third column is 633. So all the three compounds that has come from 6.3 band, and now they have resolved into three bands. All the three bands showed anticandidal activity. Again, we gone for further purification, and each of the bands, so 631, 632, and 633, they were individually scraped from the chromatogram, and then we analyzed using the HPTLC. This is the HPTLC analysis, and this is the HPLC analysis to see the purity of the compound. And in HPT analysis, we got a single band. And also in the HPLC analysis, there is only a single peak. So that means this compound has been completely purified. So the compound number 631 has been completely purified, and it has a retention time of 9.252. And similarly, there is the second compound, the 632, and that was also scraped from the previous chromatogram, and it was re-chromatographed. And again, we found a single band. And in addition to HPTLC analysis, we also performed HPLC analysis. And again, you got a single band with a retention time of 3.657 minutes. And the third band 633, which was also removed from the previous chromatogram and was run on another TLC plate. And again, it appears a single band. And so this was also subject to HPLC analysis. And we got a single peak, 3.657 minutes. So now we performed anticandidal activity with each of the compound, and we got a pronounced anticandidal activity against one susceptible and one resistant candida albicans. And this compound now has been subject to further analysis. So one of my PhD student is working very hard to get this compound in higher quantities, because the amount of a purified compound we got now is only little quantity, but we need more and more quantity, and the candidate is working very hard to get this compound in higher quantity. So once we get the compound in relatively higher quantity, that compound will be subjected to various physicochemical analysis. And finally, we go for structural elucidation. And after that, we also wanted to go for the mode of action. These three compounds show against Candida albicans. So these are all the work that we have done in our laboratory in the development of anti-candidal compounds from plants. And one particular information I failed to inform you, that we have taken several different plant species at the initial step and screened all of them for anti-candidal activity. About 2 dozen of plant species were screened for anti-candidal activity. And we found that this particular plant called Euphorbia hirta, which is a common weed normally found in all kinds of agricultural fields. And fortunately, we observed relatively higher anti-candidal activity from this particular plant. And so we wanted to purify it, and we have completely purified the compound. And the next thing is we have to go for structural elucidation and mode of action of this compound. And so here we are. And so I would like to thank the University of Madras for giving me this opportunity to work with Candida albicans. And then this research work was supported by a program called RUSA 2 program. We received a grant from Ministry of Human Resource Development, Government of India. And so she is the candidate, Smija. She's a PhD research scholar working under my supervision, and the entire work was completed, performed by Ms. Smija. And with this remark, I would like to thank you everyone for your patient listening.